Hi everyone, my name is Alejandro Salinas-Medina. I'm a PhD candidate at McGill University, and today I will be presenting a computational framework that integrates multimodal high-resolution data to simulate brain region activity. My hypothesis is that the integration of high-resolution data will enable the construction of accurate brain microcircuit models that will enable realistic simulation of brain regional activity. For this, we need to develop a scalable framework that is able to integrate multimodal data, and also we need to develop a study case for testing out the simulation of this framework. So in this specific case, I have been using data from the hippocampus because it's the region that it's more characterized recently. But this framework is scalable to any other region of the brain. And also, we need to create a validation pipeline in order to compare simulated data and the real data in order to get a fair comparison on how realistic is our simulation. So this is the main flow of the pipeline. We have our multimodal high-resolution data, and we integrated it in a connectivity mapping pipeline where we map a BigBrain surface mesh to connectivity data on a vertex level and sub-region level. Then we import this high-dimensional multimodal data within the virtual brain in order to leverage its capacities for running simulations and testing different neural mass models, different integrators, etc. After that, we have our brain region activity simulations with approximately 80% correlation with real data. And then we feed this to the validation pipeline. That we have two sub-validation pipelines. One is the graphical validation pipeline, that mainly is the comparison between the graphical comparison between the real data and the simulated data for the phase state plots, some frequency bands, expected behaviors within the hippocampus, and the presence of hippocampal spindles and barks. And as part of the validation pipeline, we also have a quantitative validation that loads the real data and the simulated data, filters this data for different frequencies, and calculates the magnitude spectrum correlation and compares it for each frequency band. So we have the targeted connectivity extraction pipeline that it helps us mainly to extract structural connectomes, and these connectome matrices will be used as the global connectivity for the simulation. So it's a pretty standard process that includes preprocessing of T1 and diffusion weighted images, computation of tactography, the transformation to native space, the reconstruction of structural connectivity matrices. So all this pipeline is actually available in eBrain, so I took it from there. And then we have the target connectivity extraction, that in this case the hippocampus connectivity is the target data that has been extracted. So we can see the different structural connectomes that have been used as global connectivity for these simulations, for these regional simulations. We see it for a healthy subject and for 200 unrelated healthy subjects. So they are young adults. We can also see the comparison of the track length, also for a single subject, and for 200 subjects. So that was only for the global connectivity, and for the local connectivity, we use our surface mesh. We create a sparse matrix by finding the shortest path for each pair of vertices in the surface mesh using the geodesic distance. And we have the surface mesh that is extracted from BigBrain, and it has 32,000 nodes. This means that our brain-region-specific network model, it's a graph of 32,000 nodes that within each node there is a neural mass model interaction. We have vertex-level region mapping and also connectivity mapping for the surface mesh. And here we can see the distribution of the edges and triangle areas of the mesh. So the narrower the distribution for these simulations, the better. So we can see a narrow distribution there. We also have a pipeline to get the real data for the comparison. So this data is available in the work by Kels, and we have the diagram flow there. And now I'll proceed to the results. So firstly, I'm presenting the brain activity simulation for a Zetterberg-Janssen neural mass model for a 1 minute simulation time running. And we can see a zoom in for 2 seconds in order to appreciate the waveform. And also, well, the virtual brain makes it easy to compare different neural mass models. In this case, I also compared the DECO1 neural mass model that, as you can see, it's also a minute long, and we have 0.2 seconds zoom in to appreciate the waveform. Now let's get started with the graphical validation results. So the first one is to compare phase state plots of both the simulations and from the real data. From the literature, we know that EEG within the hippocampus usually has fixed point attractors and limit cycle attractors. So this is the case for both simulations and for the real data. We can see the likeness of the attractors for each of the data sets. The next one is an intermittent delta activity that it's considered a marker for hippocampal intracranial EEG. So here we can see that there is some spikes on the delta activity for the real data set. This indeed is intermittent. We can observe the same pattern for the Zetterberg-Janssen simulation and for the DECO1. We can see some of peaks intermittently in delta. And delta is within 0.5 and 4 hertz. And we have the same expected behavior in hippocampal EEG for theta activity. That theta activity band frequency is between 4 and 8 hertz. So for the real data set, we can see some of the spikes for theta. We can see the same behavior for both simulations. The next one is a consistent presence of beta oscillatory activity, that this is between 13 and 30 hertz. We can see some kind of more consistent spikes within the filtered data. Well, the red line is a dynamical statistical defined threshold. We can see the same pattern for, similar pattern for the simulations. We can see it's a consistent presence of these spikes within these frequency ranges. The next one is occasional presence of gamma activity. Gamma activity defined between 30 and 100 hertz. So we can see them like randomly, occasionally appearing in the spikes in the real data set. We can observe a similar behavior in the simulated data sets. Next, on the graphical validation, it's the presence of hippocampal spindles. This is waxing and waning sinusoidal oscillations that oscillates between 12 and 13 hertz. And here in red, we can see the spindle events. This is for real data, and this is the result for the simulated data. Finally, for ending the graphical validations, we have hippocampal bugs that are high amplitude 14 hertz spikes. We can see them marked in red in the real data set, and we can see the presence of them in both the simulations. And we can see a graph for the quantitative validation results. Here we are seeing the mean for different sensors in each frequency band. We can see that every sensor has at least 0.7 correlation between the simulated and the real data for each band. And there are some sensors that are approaching to 0.8 correlation. So, to conclude, by integrating multimodal high-resolution data, we have observed a significant similarity between simulated and real brain regional activity. This all integrated within a computational framework. And in order to enhance the reality of these simulations, the development of a structural connectome based on hippocampal single neuron projectomes is in progress. That's it. Thanks.